This presentation will show the link between meiosis and genetics. Here we have a slide that shows one parent, homozygous dominant, and in metaphase 1 the homologous chromosomes have lined up side by side, big T, and big T, big T. Notice that each chromatid has already been copied in the S phase of the cell cycle. So there are two chromosomes, two strands of DNA, each of which have the trait for tall pea plants on it. Below are other chromosomes, homologous chromosomes, which have lined up side by side, but they don't have any traits on them that we care about. So they don't have letters on them. But remember, all the homologous chromosomes line up side by side in metaphase 1 and then are pulled apart in anaphase 1. Here we've eliminated the extra chromosomes, which don't have any traits we care about in this case, and we still have our homologous chromosomes lined up side by side, and the big T's represent the trait for tall pea plants. Here they've separated, moved farther apart, like they would in anaphase 1. The chromatids are still connected. They need to move apart now. In the second division of meiosis, the chromatids move apart, and here we have them moving apart, and then a new cell forms around them, and so we end up with 4 gametes, 4 sex cells, each of which have the haploid number. They have 1 chromosome for each homologous chromosome. So now if we line these up and we look at a homozygous dominant parent with the traits separated and a homozygous recessive parent, we would be crossing big T with little t. And traditionally we eliminate the actual strands of chromosomes and we just look at the alleles or the traits that we're interested in, remembering of course that these are sitting on strands of DNA. But since the strand of DNA is not that important at this point, we can take those out and just go with the traits. And I've made them different colors just to make it easier for you to see. There of course are no colors or anything to do with alleles. Some alleles deal with color, like eye color, hair color, flower color, but that's not what we're talking about here. Just wanted to make it visually interesting for you to see. So a typical cross, homozygous dominant, homozygous recessive, it's the classic P1 cross. So if we take those then and we put them on a Punnett square for mono, one single trait, mono cross, you would take each of these gametes and put them on one side of the Punnett square. And for the other parent, we would put each gamete on top of the Punnett square in one place, because each of these T's would represent a separate sex cell, a separate gamete. So you put these gametes in, what could we possibly get? What are the possible ratios of offspring? So imagine a family that has red hair in them. How many children would you expect out of 50? It's probably too many for humans, but out of 50 or so, how many children do you expect with red hair or something like that? A Punnett square allows us to get the ratios. It never allows us to predict the next child, the next offspring. For example, we know that gender is 50-50, boy-girl. So if you had 3 girls and then you were going to have another offspring, there's still a 50-50 chance it will be a girl or 50-50 chance it'll be a boy. The previous offsprings have nothing to do with that potential cross. So a family could have 10 boys, another family could have 10 girls. And that's just the way it goes. But across the whole population, you're pretty safe to say that's going to be about 50-50. So putting these down, and then what you do is you put one little, in this case green T, in every box below its spot, its slot, and one green little T in every box below its slot, one big red T in every box to the right of its slot, and one red T in every box to the right of its slot. And then it fills in, and we should be able to see that they're all heterozygotes, which means in the case of pea plants, they would all be tall, but they would be heterozygotes. And again, this is another visual of the same thing. The T's come down into their slots. These little green T's come down into their slots. You fill down and you fill across, and you get what you get. And then you take a look inside this box and you analyze your results. These are the ratios or percentages of the offspring. It could be 4 out of 4, it could be 400, it could be 4,000, whatever it is, 250 offspring. These would be the ratios you would expect from 250, from whatever. And in this case, they would all be tall and they'd all be heterozygotes.